All right, thank you for the introduction. I'm here with my advisor, Dr. Seth Murray from Texas A&M University, and I promise I will explain the title later in the presentation. So senescence is a term that describes the orderly deterioration of biological function, and in a crop like cotton, it's particularly important for fiber quality and yield, and in cereal crops, it's arguably even more important for grain quality. The identification of a fast senescing genotype in cotton could have relevance for the organic sector in particular, where defoliant usage is discouraged or not allowed. And, identifying one with divergent temporal phenotypes could also enable us to ask more basic biological questions, such as the molecular genetic basis of senescence differences between genotypes. So like many breeding traits, as everyone in the room knows, it's a very laborious trait to collect, particularly due to the revisit time that's required to obtain senescence data. And so the advantage of using a drone or a greenhouse is that we can repeatedly score senescence after the fact using both visual scores or vegetation indices. And so this picture is meant to convey the throughput with which you can collect senescence. So each individual square here is a single cotton plant. And you can track the senescence across time, moving from left to right, according to days after planting. So this project takes data from two different experiments that were conducted in College Station in April to September of 2023, and I'll refer to them as E1 for experiment 1 and E2 for experiment 2 moving forward. So experiment 1 was a panel of 48 genotypes, all of which had a Gossypium hirsutum background. These are BC5 advanced backcross inbred lines. They all contained small portions of the wild species, Gossypium tomentosum. And this was a mixture of chromosome substitution lines, or CSLs, chromosome segment substitution lines, and then 2 pure elfin lines, as well as a Texas marker 1 check. And these were 48 single-space plants. There were 5 blocks, so this was an RCBD with 240 single-space plants. And then the outer rows and end hills were just there for experimental filler to absorb environmental variation. And so the great thing about working with CSLs and CSSLs is that we're operating under a common genetic background, so the Gossypium hirsutum background, and we can pinpoint fine regions of introgression. So in theory, the phenotypic variation should be due to the introgress segments that are coming from the wild species. E2 was very similar, just consisted of a smaller panel, 27 genotypes replicated 9 times. These are again advanced backcross inbred lines, again with a Gossypium hirsutum background. But this time the introgression segments came from Gossypium mustellinum. So we followed a standard phenotyping pipeline using an RGB drone, and we were able to get 14 late season flights that we used to stitch those images into orthomosaics that were used for senescence processing downstream. So with those orthomosaics at each of the 14 time points, we created single plant shapefiles, and that allowed us to track senescence for each individual plant within each genotype at each of those time points. And so to first collect senescence data, I implemented a very simple visual scoring system, which would be typical with a breeding trait. So this was a 0 to 5 scale, so 6 different categories. And combining E1 and E2, that left us with 473 plants that survived until the end of the season. And multiplying that by 14 flights, we were left with an initial data set size of 6,622 visual senescence ratings. And I'll abbreviate those as VSRs moving forward. So while I was scoring senescence, as we've mentioned, the annotation is the most painful part. I did start to take notice of divergent temporal phenotypes. So if you can look in panel A here, the plants senesced to an irreversible state of senescence. They didn't make any sort of recovery. But B and C represent the fact that cotton is a perennial, and so it can actually have indeterminate senescence at the end of the season, which is why defoliant is commonly applied. So in B, we can see that it senesced to about a 2 or a 1, and it maintained that until the end of the experiment, whereas in C, it actually senesced partially, but made a recovery toward the end of the season. So as many of you know, every time you capture an image, it's actually decomposed into its component bands. So in the case of an RGB image, we have red, green, and blue to work with. And so I looked at 34 different vegetation indices and assessed them for pairwise correlation with the visual senescence rating, because I wanted to try to find an empirical replacement for the visual score. So the two indices that emerged on top were the red chromatic coordinate index, and its formula is given up at the top right, and then the transformed normalized difference green and red index, which is TNDGR, which actually had the highest correlation, although it was negative in magnitude. So I chose to work with those moving forward. So the issue, regardless of the type of senescence metric we're working with, is that the data set is noisy and discontinuous. And so you can see that on the left, we have the visual score and then the two different vegetation indices. And regardless of which you're looking at, it's not a true representation of the underlying process. So we want to find a way to recover the smooth underlying phenotype that actually makes up senescence, because it's not happening in a discontinuous way. And so the tool that we're going to use for that is functional principal component analysis. And here's the formula for that. So this is, in theory, we're decomposing a function into a set of basically the mean function plus a set of deviations from the mean. And so it's, in practice, we're going to keep a very limited number of principal components, so functional principal components in this case, and that's indicated by k. So we've gone from an infinite sum, and we're retaining just k functional principal components. And so again, this is the mean function for each plant. So the ith curve would just be the individual cotton plant in this case. And we're representing that as the mean. So that's the average temporal senescence trajectory. And then the functional principal component score for each individual plant. And then the eigenfunctions are the overall deviations from the mean. And then the scores weight each function for each plant. So it tells it how much of a deviation is occurring from the mean. So one of the great things about FPCA is that while we're recovering the underlying temporal phenotype, we can also reduce the dimensionality significantly of our function. So this is just what the raw data looks like for an individual plant. So regardless of the metric, we have 14 scores for each time point, for each plant rather. And then after we perform FPCA, in this case, we're going to retain the first and second functional principal component scores. And again, these are the Greek letters xi in the expansion, and those are just telling it how much to deviate from the mean function for that specific plant. And if you'll notice, FPC1, regardless of the metric, is a few orders of magnitude greater than FPC2. And the reason for that is it explains the vast majority of the temporal variation. So if you look, FPC1 for the visual score is explaining 90.2% of the variation, and then it moves down to 83.5%, and then 87% for TNDGR. So regardless of the metric that we're using, we can essentially categorize the senescence trajectories into temporal categories by using, basically sliding along the x-axis on FPC1. We can obtain plants that are undergoing a stay green phenotype or senescing rapidly. And so I've denoted that with an open or a closed circle. And you can see that RCC is a lot messier than the visual senescence rating, but TNDGR does a much better job of clustering. And in the case of TNDGR, recall it was a negative correlation, so that's why the open circles are over on the left side. So we were able to recover the smooth phenotypes for senescence for each individual plant. So this is just all of the plants in the experiment shown on the left here. And then I pulled out a few specific plants and colored them by genotype to show that there may be a sort of genotypic component that's regulating senescence. And of course, in the rapid senescence case, there were a few outliers, but 4 out of 5 replicates in panel B show that there's a genotypic component to the senescence trajectory. So the next question I wanted to ask is, can a deep learning model learn the same temporal patterns just using the images themselves? But the issue is that we have a dimensionality mismatch. So we're treating the first functional principal component score as a reasonable summary of senescence, again, because it explained between about 84 and 90% of senescence, but each plant still has 14 different images associated with it. So we have a mismatch there. And so what we're going to do is, again, we're going to use the array structure of an image to our advantage, and we'll separate each image into its component bands, and then we're going to stack them according to date. So basically we're taking each image at each time point, and we're concatenating it along the third axis. And so we basically have an array that goes from the XY dimensions times 3 to 3 times 14, which is 42. So we have an XY, Z, and our Z-axis has now accommodated the temporal aspect of the images. So we're representing them as a time series image. So this is the recipe to make the image sandwiches that I promised you. And again, the dimensionality of the image set is compatible with the FPC1 scores because every single plant has a single time series image to match that FPC1 score. And so I was very aware that the data set size is not in the millions for this experiment, so I underwent some data augmentation and essentially boosted the data set size to around 2,300 time series images. And I'd like to emphasize that although we've reformatted the input size, we haven't lost any of the spectral data because we've simply increased the depth of each image, but we haven't removed any of the time points. So before I trained this model, I got very lucky and ran into a paper that discussed hyperparameter optimization. And so I found a Python library that specifically discusses this called Optuna. And essentially you can take a subset of your data and give it a set of hyperparameters to search through. And so I modeled it after that paper that I had read, but essentially you can give it these choices. They can either be categorical or continuous distributions for something like dropout or learning rate. And you can empirically determine the hyperparameters that are going to work best for your model instead of doing it ad hoc or just using the defaults. So again, I wanted to continue to combat the modest data set size and take an aggressive train test split. And so I used the experiment 1 data set for determining the hyperparameters, and I used a 50-50 train test split with only those images. And then for the final model training, I asked it to output all of the E2 time series images using, again, those hyperparameters that we got from experiment 1. And so in the final model evaluation, I asked it to output all of E2 images, and then I did an 80-20 train test split with a 20% internal validation for experiment 1. So in total, I looked at 6 different models, and M1 through M3 are assigned to the visual senescence rating, RCC and TNDGR, and then M4 through 6 are just copies of that, but the difference is that I allowed the dense layer to be searchable by that hyperparameter optimization framework. And so the models are very similar, but I was just curious if the specification of the dense layer would affect the outcome. So another great thing about the hyperparameter optimization is that it doesn't just give you the parameters, but you can actually get the variable importance of each parameter. And so it's kind of looking into the black box in a way. And so as we expected, the learning rate had the most important outcome for visual senescence rating and RCC, but interestingly, that was not the case for the transformed normalized green and red index, where other features such as the number of layers and the activation function had an influence on the outcome. And so to me, it was worth it then to do the optimization because there's no one-size-fits-all set of hyperparameters. Okay, and looking at the metrics for each model was evaluated 25 times with a randomized train test split. Based on R-squared alone, we can see that the visual senescence rating performed the highest with a very low mean absolute percentage error. M1 performed very similarly. In second place, the TNDGR index did pretty well with a 0.74 R-squared, also just below 6% for the mean absolute percentage error. And RCC did not do as well. And that didn't surprise me again, because if we look back at the FPC1 versus FPC2 plot, we could see that RCC was not able to separate those temporal phenotypes as well as the other index was. So you can actually treat the functional principal component scores as a data set. So a lot of times people will do multivariate, or in this case, univariate analysis. So I kept the FPC1 score, and I actually looked at analysis of variance for both the actual FPC1 score and then the predicted score from the deep learning model. And we can see that there is, in fact, a genotypic component to the temporal senescence trajectory, at least in this panel of CSLs and CSSLs for cotton. And interestingly, the repeatability was actually higher after using the predicted values from the deep learning model. So I generated saliency maps and this is indicative of all of the images in the stack. And we can see that, at least for the top performing model, the model is looking exactly where a human scorer would look. So it's encouraging to see that throughout the temporal domains that we added, the model is looking at the appropriate pixels for making its predictions. So again, to wrap up, we were able to use FPCA to significantly reduce the dimensionality of our time series data set while simultaneously recovering the underlying phenotype. We found candidates for replacement of visual senescence ratings with those two indices, although TNDGR did substantially better. The regression models were able to, in most cases, map back the time series image stack to the FPC1 score, which was a summary of our temporal senescence phenotype. And then looking at analysis of variance, we confirmed that there was a genetic influence on senescence trajectories. And I would argue that FPCA can serve as either a basic or an applied tool, because you can actually look at the temporal phenotype as opposed to evaluating phenotypes at static time points repeatedly. So there were definitely some limitations to the approaches that I used. For example, it was pretty computationally intensive to run a lot of Optuna trials because I set so many different parameters to search through, so we may not have converged on the optimal parameters. There was, of course, noise inherent in the drone images, and that could have reduced the regression model's ability to map back the pixels to the FPC1 score. And this was these were simple convolutional layers, and so perhaps using LSTM layers or gated recurrent unit could help with more complex models. And in addition, this data set didn't have any sparsity in the sense that every plant at those 14 time points had an image, but it would be good to test this by removing a certain percentage of images at each time point to test its robustness to missing data. A lot of people went into making this work possible. I'd like to thank my advisors here today. I'd certainly like to thank the Stelly lab for letting me use their germplasm, and I'm very grateful to all of you for paying attention. So thank you for your time.
Thank you so much, Aaron, for presenting your excellent research. Questions for Aaron?
Hi, I have a question about the image sandwiches. So from your slide, I understood that you're stacking these 2D images on top of each other across the different time points. Do you think that maybe perhaps the order how you're stacking the different channels has some kind of an effect on the result or the modeling? I mean, maybe putting all the red channels next to each other or something like that. What do you think about that?
Yeah, it's a great point. I guess the default loading in, at least in TensorFlow, has red, green, and blue. So it would be very interesting to see if blue, green, red actually makes a difference. I honestly don't know, but it is a good idea to test that. Thank you.
Hi, here. So from what I understood, first, it was a great talk. You did the sandwiches by stacking RGB and then the time points only in the channels. Have you tried using 3D tensor? You know, you have channels, X, Y, and Z, and then stacking on Z. So you do 3D convolution instead of 2D?
No, that is a good idea, though. I appreciate the suggestion. It was just 2D convolutional layers, so
yeah, in that way you can use the channels as they are, and you use the same idea, but you have the Z axis to do the time steps, and then you can do the regression. So the channel order won't matter.
Okay, thank you. I appreciate that. Sure.
Over here. So what network architecture did you use? You mentioned some other ones to use, but what convolutional based architecture did you use? Like recurrent neural networks, or?
It was strictly convolutional. I didn't actually use LSTM or GRU or anything like that. It was strictly a convolutional net.
Did you use a ResNet architecture? There's lots of different architectures that you can use that can provide different benefits did you explore different architectures? The
No,
GRU and
yeah,
those
yeah.
are very, very different than convolutional based.
No, I understand. I did not pre-train the model. If that's what you're asking, it was strictly trained from scratch.
Okay, thank you.
Hi, great presentation. Did you account for background in the images? Like, I don't know if the soil would be a factor in this, or did you binarize it such that you're only analyzing the plants, not the background?
Yeah, it definitely would affect it. I had issues with I had issues with masking out the soil because the senescence phenotype almost looks identical to the soil. So I would like to go back and redo everything with masked images, but that there definitely were contaminating soil pixels in there. And the activation maps show that in some of the other models.
Aaron? So I have one question, Aaron. So how you made sure that senescence is just because of nature or naturally, not because of any disease? Because the crop I work, we see this kind of senescence a lot when we have these fungal diseases, leaf blight and leaf spots. So.
Sure. Well, I visited the field to confirm that it was senescence, but it's certainly possible that I missed out on a disease in that particular field. It's known to suffer from blights and things like that, but I'm pretty confident that it was natural causes. Yeah.
Excellent.
Yeah, okay. Sure. Here. So use RGB, but I think if you would use an infrared channel next to the RGB, you would get an easier soil plant segmentation. And I think if you would put the NDVI only in your training network, it would be more straightforward. Now you're not sure what your model is actually using as a determination of the senescence level.
Yeah, I agree with that, and it was just due to I kind of improvised that experiment last minute on somebody else's data set, so I did fly this summer using a multispectral drone, so yeah, I'll incorporate NDVI this time around. Thank you.
So if we have no more question for Aaron, let's thank Aaron for giving excellent talk. Thank you.